Okay, so everybody ready to get started for today? Go over the quiz a little bit, see if there's any questions about that kind of stuff, and start transitioning forward to today's material. So today we're going to do a handful of stuff. We're going to blend together some theory things. We'll talk about some aspects of filtering and some theoretical aspects so we can understand what's happening in the data. We'll work with some data, and hopefully we'll get a chance to actually show you guys how we collect some data as well. So we'll get to that a little bit later. First, just a couple notes. Let's see here. So I know that we're practicing writing in this class and scientific writing, so I just wanted to remind you guys that that is a hard thing to do. Scientific writing is hard. It sucks. It's always hard. It never really gets easier. So I say that not to be intimidating about it, but actually to hopefully mitigate something that can be intimidating about scientific writing, and let you know that it's something that we all go through as a learning process, and it never really stops learning either. So I'm working on a scientific manuscript right now with the dean, and you should see the stuff that I have to correct that he writes, and vice versa. So it's always this exchange back and forth of constant revisions and editing of each other's work, because iron sharpens iron, right? Steel sharpens steel. So you have other people that help to strengthen what we do, and we don't get it right the first time or the third time or the 10th time. It takes practice, and so that's a normal part of the process. So don't get discouraged if it might seem like it's struggling a little bit. You're at the very beginning of your career, so it's going to take a little bit of rust and dust to get a little fluent with it. And like I said, I've been doing this for 15 years, and I'm working with a dean who's been doing it for 40, and neither one of us send each other really nice drafts of things. We both need to revise and work on each other's stuff, and it's a helpful process. So hopefully that provides some context for you too. So don't take it too hard or anything like that. If there's aspects that struggle, that's a normal part of growth. So that's okay. We'll talk about some artifact rejected trials. Hopefully you guys had a chance to work on that a little bit through the week. A couple points to note about that. I know a lot of people sent me emails yesterday. They said they had some different types of software snafus, or some things were working, other things weren't working. It's a really good point to remind people of how useful it can be to actually get to those things Monday or Tuesday, because we only meet once a week. So try not to do this stuff Thursday night, because if something goes wrong on Monday or Tuesday or Sunday, Ariel and I are much more readily available, and we can sort things out and give you that extra time in the week to actually really spend some good time being able to work through it. Because sometimes it's just a little weird error that goes wrong that might take hours and hours to fix if we're searching around in the dark. But if you can be like the Who Wants to Be a Millionaire show, phone a friend, we might be able to sort that out for you in like 5 minutes and save a lot of trouble. So, be friendly and shoot us emails, ask us questions if there's something that's going wrong, because we can save you guys a lot of trouble, and efficiency in that case will be improved. So we're here to help you guys, and we want to do it. And so try to get that stuff done early, I guess, is the take-home message there. And so we will. We're going to keep working on those methods and results and introduction sections as we move along. Hopefully you guys had a chance to work through the artifact rejection process, practice working and basically identifying what some of those artifacts were in the data, and see how that process goes. Get some time practicing being fluent, just working with the MATLAB environment on your computer, working with the data, see how things change, see how things go. So yeah, you said you were able to do a lot of that, right? How did that, by the end of it, was it a lot more fluent than the beginning? Yeah, it's actually pretty straightforward once you kind of clear those hurdles, right?
Yeah, just like kind of knowing how to do it.
Yeah,
That's
so
the only, everything else is pretty straightforward.
it is definitely gets to be annoying, and so remind me to come back to that point, because there's a couple things I want to mention about that. But since we're talking about artifacts, maybe it's a good time to touch base on. We'll just cover some of the aspects of the quiz that we just worked through today, because it actually talks about some of the artifacts. Okay, so I'm going to write a note up here to come back to the annoying part. Okay? So this is going to be like right over my head, which is like labeling me as annoying, I guess. But try to dissociate from those two things, me being next to the annoying thing. Let's talk a little bit about the quiz. Okay, so the first question we had is, if you wanted to create a difference wave between ERP conditions, what text file would you use? Would it be the events list or the bin operations file? And then accordingly, which of those text files is used first in the normal sequence? I'm sorry?
The bin operations first and then the event list?
Yeah, so, well,
I mean, the first answer would be bin operations.
yeah, so there's sequentially in the terms in the way that the question was asked. First question: if you want to create a difference wave between two ERP conditions. So if this is our standard, let me see if I can get another marker in here too. I'm gonna do all my markers. Oh, there's some down there, I guess, huh? Let's see. What's up? So we have our ERPs, right? And you have, huh? You guys good? Okay. Oh, yeah, question. If you wanted to create a difference wave between these two ERPs, let's say that this is the ERP for new items in a memory test, and this is the ERP for old items in a memory test. What would you do? Or what do we say? What is the text file you would use to create that difference wave? Yeah, bin operations is the idea for that. So bin operations, what you're doing is you're taking, let's say that we end up calling this, let's say this is bin 5, and let's say that this is bin, I don't know, 10. What you would do in a bin operations file is you might write, excuse me, I might sneeze in the middle of this, but don't you hate when that happens? You might write some a code in the bin operations text file that might look like bin 80 equals bin 5 minus bin 10, and the label for that would be old minus new. Okay, so we have 4 parts to this. We have defining what our new bin is. We have defining what the calculation is that's being performed. We have this marker here that tells us, well, what are we going to call it? And then this is the answer to what we're going to call it. So you have those parts. Now, what's a difference wave?
Just to subtract it.
Yeah, you're subtracting this from this. So what would it look like if we plotted the difference wave? This is kind of it's going to take some mental gymnastics because we haven't talked about this yet so much. If you plotted the difference wave of these conditions, what would it look like plotted up against here? All right, Tom, come on up and show us. Can you show me? Yeah. My hand hurts sometimes. I can't draw, you know.
I'm guessing it'd probably look like something like this.
Okay. Is that what you guys had in mind?
No.
So, what is this? What is the green line that you drew supposed to be representing?
The difference between the...
Yeah, the difference between them, right? So, is there a difference between red and black here?
On this one, not right here, but...
Then why is the green line going up?
Well, unless they're absolutely touching, which I don't know if they are. But I see the spaces here, so...
The value between the spaces like 0 to
Yeah.
so it should be flat.
Yeah.
Oh, okay.
Let me show you how I would fix it up, because how it would actually look. It would be... Yes, you can. So, this is what our hypothesized one was initially. And it's not exactly correct, and I'm going to try to show you why. So what I'm first going to do is I'm going to get rid of it so it doesn't distract us, and then we'll replace it. Because remember, this is actually really a helpful exercise to mentally conceptualize what a difference wave is. You able to see back there? Okay, cool. So let's say that this green marker is our difference wave. Now remember, this is the subtraction. Remember we went back a couple lectures ago, we talked about the LPC, the FN400. They are the different, when you plot the topographic maps, the big 3D color heads, those are representing the subtraction between this. So this might be the FN400 here, right? This difference between here and here. So you're subtracting these two. Or basically, it's another way of thinking about it, saying, show us everywhere where this black line, or the old condition, is greater than that in the new condition, is another way to think about this. So if we would plot that, right, this would be just a tiny little blip. It's pretty much nothing. And then it's pretty much at 0, right? So this is our 0 microvolts. It's all the same, the same, the same, the same, the same. It starts to get a little bit bigger. Right? And then it gets, so now this is our biggest point, so this is our peak, and now as I come up here, these start getting smaller. So now, as this comes here, and now we're back to this point. Yes?
Are there ever like negative values?
Oh yeah. Yeah, so let's say, so here, right? So this is convenient. So now it flips around. So here we had basically black greater than red, old is greater than new. But here, now we have basically new greater than old, and so if we continue our black line, excuse me, our green line out for our difference wave, then this would actually start here. It would start to come a little bit less negative. Yeah. So for me, that when I was learning this stuff, that graphical representation really dialed things in into what a difference wave represents. Yeah, you guys follow? Cool. And it's, if you notice, it's pretty substantially different in terms of how it's how it looks from our regular ERPs. And so that's the idea there. And the second part, of course, you would use events list as a text file first, because our order of operations is, right, we basically, we're loading in the data. What do we do after we load? So we've already loaded in data that was basically, we loaded in the raw data earlier. We processed it so that it had a high-pass filter, 0.1 hertz. Then we applied the downsampling from 1056 to, or 1024 to 256 hertz. What do we do next? What do we do basically? So we had our order of operations. So we loaded the data in. We already loaded it such that we had re-referenced. We did high pass filter. Then we did our downsample. What happened after that? What was our next step?
Event list.
Okay, yeah. So here we did our event list. Okay, so this is that second part of our events list. .txt. This is the second part of that first question. What are you going to do first in the text files? Are you going to do events list or bin operations? And the answer is events list, because what we're doing with the events list structurally within the computer code is we are first creating a list of every single possible event that we talked about before class. Remember that? And then we give a label to each one of those. That's where we're sort of like sorting the socks into different bins. Like red socks go in this bin, blue socks go in this bin. White socks go in that bin, whatever, and we can categorize based on conditions. And then, what do we do after we create our events list? Yeah, we epic the data. We turn it from one long string of EEG into 300 discrete little chunks of every unit. You guys remember? So here, and we'll epic the data. Okay, what do we do after we epic? Yeah, you're going to artifact reject. Hopefully you guys spent a lot of time practicing that for homework this week. Artifact rejection. What came after that then?
Is that whenever we created the average ERP?
Yeah, you want to create an average ERP. So now we make that fundamental shift from EEG data to ERP data, right? So we stop using the menu in EEG lab and we start using the menu in ERP lab in our GUI. You guys remember how we sort of switched there? So now we're going to create average ERP, and that's going to give us a .erp file output. God bless you. Now, what did we do now that we have an ERP file as opposed to our EEG file? Yes, now we're back to that last part of our first question, bin operations. So now we apply our bin operations file .txt, which is a text file, which is exactly what we explained with that example there. Okay, so structurally to the first question, you're going to apply the events list first before you would apply bin operations. And if you try to do it reverse, the computer's going to try to throw up on you and give you some ugly red error codes saying, we can't do this. Make an events list first. And then you make it, and it's like, all right, sweet, thank you. We'll do bin operations now. So it's important to know these structural aspects of where we're going in our sequences, because you're going to be expected to be able to do this independently on your own. Like that, this will be part of the midterm and things like that. You're just going to be given data, and you're going to say, it's going to say, run with it. Tell me what's the answer to this condition at this time at this place. What's the statistical answer? So, and you'll be up there. We'll get you ready, but you should be able to know this procedure, and it'll come really easy. It just comes with practice. So we'll talk later on today about what we're going to do after this and after bin operations. We'll talk about how we visualize the ERPs a little bit more, how we obtain some of the statistical measurements for them, how we plot them, and come to some of those conclusions and things like that. But that's the idea there. Taking a step back more broadly, the second question asked what the inverse problem was in ERPs. So we talked about this. This question basically came directly from class, last lecture period, but if you weren't able to make it, it also was covered in a handful of our text and some of the article. Basically, it's a pretty standardized issue in ERPs, and so you should have been able to pick it up if you hadn't been able to make it in some of the required materials. So anybody remember or have questions about what the inverse problem was? What was it, Tom?
Would mathematically possible to update the neural generators just by looking at it from the scalpel?
Exactly. That's it. It is mathematically impossible to know exactly where in our brain a given pattern of activity at the cortex can be localized to some type of subcortical location, because there's an infinite number of possible solutions of neural organizations of different patterns of neurons that could theoretically and mathematically give rise to anything you see at the scalp. So it's impossible to work backwards and say, we have this, can we figure out where it came from subcortically? It's just not mathematically possible. Now it is, of course, just as a point of context for comparison, you can do the forward solution. You can start with activity here, and you can figure out a mathematical way to kind of like figure out where activity here would be more likely to project up to the cortex. It's just that we can't do the opposite, which is really tempting because we want to. We want to see something at the scalp and go, yeah, it comes from here, or it does that. It's tough. Now, to be fair, there are people who claim that they can devise some types of equations and algorithms that try to address that. It's called source localization, and there's a whole little field about that. A lot of people are skeptical about it. Not that it doesn't work in its entirety. It could end up being a fairly good approximation, but a fairly good approximation is not a definitive answer, is it? So that's where people kind of come down on that fence. You know, you either think it's kind of, you guys remember The Simpsons? Is The Simpsons too old for you guys? No? Okay, I know Simpsons have kind of gone out of style lately, right? It's more like Family Guy and South Park or something. But there's a character in The Simpsons, Krusty the Clown, and he used to have this little saying. It's like, so he was famously kind of a crappy clown, And so he would endorse these products, and he would say it gets the crusty the crown seal of approval. It's not just good, it's good enough. And so that's kind of, you can think about it, source localization. It might work a little bit, but is it, or can you be absolutely sure? Not yet. But so other people, you know, you can look at it through the other lens and be like, well, not totally sure, but it gives a pretty good enough approximation. So different ways to look at things. I happen to be kind of a curmudgeon on source localization, so that's just me personally. Alrighty, enough about that. Third question, we were trying to label a bunch of different artifacts in the data. Okay, so the picture itself that we worked on came directly from Steve Luck's book, and as well as from the slides that we introduced last class. So hopefully that jogged your memory a little bit, but if not, we also worked with a lot of these example artifacts just in drawing it on the board. So we had, let's see here, if you had EEG that was looking like that, what would that have been? EMG, exactly, muscle artifact. If you had, what was our second one? Let's say we had, what was that one? EKG, right? Boop, 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 boop. Okay, that's your heartbeat. The other one, next one was, if you had, oh, we talked about this a lot before. Let's say you had this. What would that be? Yes, skin potential, drift, handful of different things could qualify for that. What if we had, instead, that it came across here and started rising and just looked like a plateau? Blocking, exactly. Now, we also had a different pattern that we kind of had EEG and then alpha waves, yeah. So a lot of times you're going to see alpha in the occipital electrode sites. They happen to do a lot with drowsiness and things like that. What if you had an electrode that was coming out this way, recording, and then it went this? Yeah, saccade. So that'd be one of the ocular artifacts. Good job. What if, and you could also have looked like, what if you had one that looked like this? Would it be a blink? It's a saccade. It's the other electrode, guys. Just the same thing. Right? Because remember, we record from both sides. So if you look left, this one's going to be going this way, but this right electrode is going to be going here, right? So it's now the same thing. So we talked. Somebody said blink a while back. So let's say recording here or whatever, right? It's going to be pretty indicative of a blink. You worked with a lot of those, and you're probably very sick of them, right? You feel comfortable, you know what a blink looks like? Yeah? Okay, you're like, yeah, we'll get rid of that. Okay, now it didn't go this way. You could see another one that went that way, right? Didn't they usually look at kind of in this paired look? Okay, why? Yeah, same idea with why the saccades went both up and down. You have one on each side. The eye electrodes, you got one actually on this side usually, but yeah, you have one up. So when you crunch your eyelids and you blink, one goes up, one goes down. So those are our artifacts for question 3. You get a point for each of them. And did I give you guys extra credit on this test? Yeah, you guys. It was said. Describe something you read this week in any of the course assigned readings or materials. Write anything you want about it, what you learned, what was confusing, what was really interesting or really cool or really dumb about it, whatever you thought. Hopefully you guys had a chance to do that, get some points back. What was something that you guys found that from your reading that you might have used for that 3 extra credit point?
It's ways.
Different ways, okay, cool. Yeah, kind of interesting way. Anything else? You guys just let that one go? You said, screw it, I don't want the extra points.
I'm grading their answers.
Oh, they're just quiet? Okay, cool. Well then, we can get done with the test. So a couple things I wanted to cover just as some points. Two things. We're going to record some data today. We'll see how that's done. In a little bit, we'll go walk over to that lab. We will. So I want to talk about two things before we get into it. One is when we record data, we don't record from the scalp, do we? So if this is our research participant. Okay, we hope that they're smiling. We have an electrode that might be recording there. Okay, a little wire that comes out to your computer. Okay. And it shows that they're happy. Okay. This electrode is not actually on the scalp. We'll see this in more detail as we see that today. It's actually recording from the little space. So we showed some figures last class and some of the slides of when we talked about some of the biophysical bases of recording EEG and how they're the summed postsynaptic potentials of those neurons, right? And we zoomed in. If this is like, we looked at a picture where we had the skull, right, and we had our scalp, and then we had essentially a little thing of your meninges, and then of course we had our cortex, right? Our cortex has different layers within it, because brains are like ogres, which are like onions. They have layers, donkey. And so we're actually recording our electrode. You guys might remember, we did have a picture like this on the slides. It just looked nicer, because I'm a bad artist. But the point remains the same. This is our electrode, and there's actually a space of air in between here. It's not actually directly even on the scalp, is it? Everybody know what we do to help make that connection then? Yeah, we're going to use conductive gel. So we'll want to introduce that today because you guys will have a chance to do that. And so why do we use conductive gel? And what is it conducting? It's conducting electricity, right? It's like a classic dance at weddings. It's the electric slide. It's electric. Yeah? Okay, so we have electricity coming through here. We need to record it. What is it that helps conduct that electricity through that little space of air between the scalp and our electrode? So remember, this is our cortex. This is where all the action's happening. Okay? This is where all the magic happens. It's like Cribs. Okay? Everything's happening here in the cortex. That's what we care about. We don't really care about this stuff. This gets in the way. This is impeding our signal. We'll call impedance later on when we record data today. So this electricity has to travel up through here and somehow get detected by this electrode. We put gel in between here, and this gel is basically a thick salt water solution. So it's just saline gel. The reason that is because the ions of salt help to conduct that electricity. So how many of you guys would take a toaster that's plugged into the wall and jump into your bathtub with it? Huh? It is plugged in.
Just deionized bathtub water.
If it was what?
Deionized bathtub.
Ah, exactly. It would have to be deionized. So water, as we typically think about it, is not distilled water. It has stuff in it. It has ions, which is why that's such a dangerous thing to do, because it's going to conduct the electricity from the plugged-in toaster, right? However, if the water was distilled, like distilled water, water doesn't conduct electricity. And to be perfectly safe, now don't do it. I mean, seriously, don't try this at home, kids. But theoretically and conceptually, the idea is that it's the ions within the water that are conducting that electricity that makes it so dangerous, that allows that electricity to travel and spread from the toaster to the water to you. Don't freak out. Okay? That's why we're putting this little bit of gel in here to allow that electricity to conduct, because it's already conducting in some ways through here, right? Now remember, this is where this is the magic, so to speak. This is where all the action's happening in the cortex. As it's moving through these substances, the skull, the meninges, the scalp, it's weakening the signal, right? Because it has to travel through a bunch of stuff, right? It's like, here you've got this, and it just wants to run real fast. And then as it comes through here, it's like having to slog through mud. It's like, it's messing up the signal and smearing it, right? And then it gets even more smeared as it comes out here. Now what's incredible is that we can still pick up and read a meaningful signal from here. But we're going to use gel, so our saline solution gel is going to do that by virtue of the salt ions, essentially. So when we put these electrodes on people's head, we need a way, and those electrodes are going to have like a little cup around it to hold the gel in place. And they're going to rest on, say, someone's head. We need a way to get that gel in there, and the way that we'll do that is we're going to use a syringe, put a tiny little needle on the end of it. It's a blunt needle, it's not a sharp one. And we just use it to inject through a hole that basically creates that channel down into. So, well, let's make this more three-dimensional now. So now there will be a hole there, and well, you guys will see it in real quick, and you'll have the syringe basically dial in that gel to spread in this cup. So that's why you'll see that we'll have some of these medical devices like syringes and things like that. It's for that purpose. It's just small enough to fit just in the right hole to fit the gel into where we need to get it so that it conducts this signal from here. Okay? And then we'll use, this is the scalp here, right? Now, ladies usually get this answer before the men do. What do we want to do to the scalp to help clean it up and make a better connection? Huh? So, yeah, that's what the guys are thinking. You don't shampoo your skin. You exfoliate it, right? And you guys like to go to the beauty salons and exfoliate. I guess what do you get charged money to do an exfoliator or something? I'm told, right? Is that true? No, am I wrong?
Yeah.
I am. What am I talking about here, Ariel?
I don't know.
You don't know? Neither do I. But there's like, I was told there's like exfoliating like face scrubs and stuff to like clear off the top layer of dead skin, right? What?
You exfoliate your face, sure.
Exactly, the skin. We're talking about the scalp here, skin.
Okay, that's a facial. That's not a hair.
So yeah, obviously I don't know what I'm talking about this stuff. So didn't I say that the ladies would know better than say I was? So the idea here is when you're exfoliating skin, right? It's not a facial or something. We have dead skin all over. All right. Now that's going to create another barrier to this electrical signal being recorded at our destination. So what we want to do is exfoliate that a little bit, as you can think of. Basically, just scrape off that layer of top dead skin cells. And so we use these little toothpicks. Kind of have little sharp edges on them, so you got to be real careful. You don't want to be poking people, but you do want to kind of agitate it a little bit. So you're going to kind of like, we'll have a little toothpick that comes in through that cavity, and we'll scrape off and move it around a little bit just to kind of agitate the skin, so you pull off some of those top layers of dead skin cells, and that helps us to give a really good connection. Because you guys just spent a lot of time, presumably this week, going through all this data and rejecting some of the artifacts that we identified, correct? Were some of those artifacts just really messy, crappy data? And could you not tell the other times when it was really clean, nice subjects? Right? One of the reasons that there can be those differences between when the data is reported is because there can be better connections that were basically had those skin sites better prepared. So you guys have seen that now on the data side. We'll look at how that was collected. Okay. So that was the first thing I wanted to mention. The other thing I wanted to mention is more on the analysis side, which was once we get this signal in this computer, right, we talked about all these artifacts that can be in there, and we have our EEG signal, and what was something that we said you could do to account for some of these artifacts? You can filter it, right? So we do a high-pass filter to get rid of basically as a linear D-trend, to get rid of some of the drift or skin potentials. We talked about other ways we can filter the data. We said a little bit about low-pass filtering. So, we got rid of our series of events, but later on when you have ERPs, right before you make your figures, you're going to apply what's called a low-pass filter. So we've already done the high-pass filter, let's say 0.1 hertz. And what was a high-pass filter again? Can you guys remind me? Was it allowed to go through?
Higher frequencies.
Anything higher than this 0.1 hertz. So if it crosses this threshold, anything above here equals okay. All right, anything below here gets cut off. Now, when we have a low-pass filter, okay, we'll filter that at, let's say, 30 hertz to get rid of the high-frequency EMG noise. Remember we said we can filter out our EMG? So let's say we do now this is later on in our processing stream, right at the very end, you're going to do, let's say, a 30 hertz low-pass, and the idea is kind of the opposite here. So anything below 30 hertz is going to be okay, which means anything higher than it in our frequency spectrum is just going to be whoops, washed out. We're just going to filter that out. Now here's the thing I wanted to mention. It's an important part about filtering. And Steve Luck's book that we have assigned dives into this in a lot of really good detail. But the important point to remember and to keep in mind is that when you're filtering data, you're distorting it. One way or another, you're adulterating the data. Our overall goal is to understand the physiology of the mind. And what we want is to have the best understanding. We want the purest type of data. Now that does mean clearing out some of our artifacts. It helps to clean the data so we have a purer signal. But we also want the data to be the least adulterated, right? We want it to be the closest to the real brain activity as possible. And when you filter data, you inevitably change it in some ways. Okay, so it requires a really healthy balance and understanding of what you're doing to the data so that you can get rid of the crap but maintain the signal. Okay, so we call that essentially signal-to-noise ratio. You want to make sure you don't throw the baby out with the bathwater. Okay, and so when the thing that, again, that I want to emphasize about filtering is that it can distort your data, so you want to do it to the minimal amount possible. Now, sometimes the distortion is good. You want to get rid of the crap. Other times, the distortion is just negligible. So sometimes, depending on the type of filter you have, if this is your normal signal, right, it might change it so that after you filter it, it might come like that. Now, it basically maintains the same general aspects, right? But it is still fundamentally changed. And that happens at every subject, and then you average all that together. So it's possible that you can change your data and sometimes eliminate effects or create fake effects if you filter too much or filter in the wrong way. Okay, so we're not going to belabor that point anymore, other than it is important to know that point of caution, know what the limitations of filtering are, and revisit that chapter in Luck's discussion about it, because it'll help clarify some more details for you. Are there questions about this stuff? That's mostly what I had to hope to cover theoretically about introducing this stuff today. It's about 11. Do you guys want to go record some data? Check it out. So the lab is over in Green Hall, so we can pack up and visit there. And there's another graduate student there who I'm collaborating with on an ERP project, Mitch Meltzer. He's great. He's done a lot of work getting this thing ready for us to come visit and check it out. And it's a very basic lab, but he's worked really hard, so it'd be really nice with him. And it's kind of a small lab, so we'll have to be kind of cozy together. But we'll check it out. Sound good? All right, let's do it.
Will you log out?
Yeah, that's probably a good idea.